just start recording here. So yeah, welcome everybody to this first ever panel discussion as part of the RCN for Evolution and Changing Seas reading group. So Katie is going to give a short presentation just on what the RCN has been doing. It's kind of getting, it's a getting towards the end of its kind of 5-year mission. So, but what we have done in the past and what this reading group has been doing in the past, and then just get you up to speed on that, and then I'll introduce our panelists, and then we'll have a little discussion. So, yeah, Katie, if you want to take it away.
Thanks so much. I'm really looking forward to this panel today as well. I'm going to share my screen. So we are the Research Coordination Network for Evolution and Changing Seas. And I forget how to play this Google slide. Is it this one? Here we go. And if for those of you that aren't aware, research coordination networks are formed to advance a field or create new directions in research, to bring investigators together from different fields and do interdisciplinary topics and come up with innovative ideas for training. So we have formed this network to bring together evolutionary biologists and marine scientists, given that there's been like a lot of historical research in marine ecology, but more recently, marine evolution research is just starting to come to the forefront. So we have formed working groups. We've done professional development exchanges. We've done these coordinated readings every spring, where we have a set of papers that we all read and discuss on Slack. And we usually have dozens of participants from all over the US and some international labs as well participating in that. And we've also run a virtual lab meeting program to bring junior people who may not be in our field to our lab meetings, where they can learn some of the soft skills of like what's happening behind the scenes in science and get feedback on their career trajectories. Some of our working groups have published reviews on integrating eco-evolutionary and oceanographic models, thermoplasticity in marine and terrestrial systems, synthesis on what drives plasticity, evolution of complex life cycles, new quantitative methods for studying gradient variation in the sea or on land. We've also have working groups that are still in progress, publishing website tutorials. So we have this marine omics page right now, which is great. It would be great to put some like things on here about measuring deleterious mutations, for example, but this is a living resource that we can update. There's also temporal genomics has been doing a synthesis and review, and we have a course website for marine bioinformatics, it's like an introduction to bioinformatics class for undergrads, but using marine data sets. This is kind of a timeline of what the network has been through. We started with coordinated readings back in 2019. We held a synthesis workshop and formed 6 working groups initially. Then we were going to have a genomics workshop, but it was canceled due to the pandemic. And the Black Lives Matter movement really inspired us to do more outreach and diversity and inclusion. So we formed a diversity committee and started this virtual lab meeting training program and junior research exchanges. And then finally, last year, we had a training and integration workshop where we brought a lot of the working groups back together, and a lot of the graduate students that have been part of the network and part of those virtual lab programs also came to that. So that's sort of a brief overview of the network and what we've done. I've got, lots more to talk about, but you can find a lot of information on our website, and we have an open network. So if any of the panelists today are not part of our network but would like to join, you can learn more about how to join here. And we would love to have you be part of the network. We also have information on current activities and working groups and readings, etc. I think Nicholas, who's on this call, helped to put together this batch of coordinated readings. This year, we're doing a coordinated readings on evolutionary interventions. So we're reading about, like, for example, genetic rescue. And in the past, we've done temporal evolution, conservation genomics, plasticity, and just sort of like a first pass, a wide variety of evolutionary papers. These readings have been really great for my lab, and I hope that we can continue to do them in the future. So with that, I'll just stop there, and I'll hand it back over to Brendan.
Cool. Okay. Thanks, Katie. So yeah, I'll give some quick intros for each of our panelists here, and then if after each intro, if you guys want to kind of just give a short introduction on your work, touching on how genetic rescue fits into that work, and how you envision genetic rescue as a component of conservation and management, what we need to know, what are the research gaps that are currently preventing us from implementing genetic rescue as effectively as we would want to. So that would be great. No slides needed. I think some of you might have slides. If you want to show them, that's fine too. Yeah, but I will start with Marty Kardos. And you can feel free to correct me if I get any of these details wrong, but Marty got his PhD from the University of Montana, and did some work at Flathead Lake Biological Station, and now is a research geneticist at the Northwest Fisheries Center. So does that sound about right?
Yeah, I threw a postdoc in Sweden in there in between, which was quite fun. Yeah, so should I go ahead?
Yes, please.
Okay. Yeah, I'm at the Northwest Fisheries Science Center now. I'm a research geneticist. I've studied a lot of different organisms. I've studied songbirds, bighorn sheep, wolves. Now I primarily work on salmon and killer whales, and I'll be involved with a much broader range of species in the near future. A lot of my work has involved looking at improving ways of measuring individual inbreeding and inbreeding depression in natural populations. And a lot of my work right now is still focused on that, in particular in killer whales. And, some of my work has sort of peripherally involved genetic rescue, but most of my work is sort of more broadly involved in inbreeding depression, both theoretical analyses, and we're just about to publish a paper on inbreeding depression in North Pacific killer whales. That should be out in a couple of weeks. And so what are my thoughts on genetic rescue as a component in conservation? I think it's a great tool a great potential tool for management of small populations that are declining or failing to recover despite sufficient environment for a population to grow. It's sort of been fantastically successful in scenarios like that. There's probably dozens of papers now reporting increased fitness after outcrossing in small populations that are declining or failing to recover. But I think this is sort of like an emergency medicine scenario where, a population is basically getting ready to blink out, and one possible management action is genetic rescue. And, I've thought a lot about the fact that the populations for which there is sufficient information and available resources to do genetic rescue is really small. So the fraction of populations that could likely benefit from genetic rescue that actually can get it, must be minuscule. It must be much less than 1%. I mean, there are probably many populations of species that aren't even described yet that could benefit from genetic rescue. So I think the sort of broader importance of all of this empirical data on genetic rescue is that it sort of highlights the importance of maintaining natural levels of connectivity in ecosystems, and sort of informs how we should manage landscapes and the importance of managing or maintaining connectivity between populations that were historically connected. What are the problems? So what do we need to know in the future? I think one question is, what fraction of populations that are declining would benefit from genetic rescue? That's an important question we don't really have an answer to yet. And identifying particular populations. So sort of understanding broadly, of all the populations that are declining and threatened, how many of those are, declining because of genetic factors? And the other side of that is identifying populations that are particular populations that are threatened by intrinsic genetic effects.
Great. Okay. Yeah. Thanks, Marty.
Sure.
Yeah. Okay. So next I will switch to Chris. So Chris got his bachelor's from the University of Chicago. Did a PhD at UCLA with Kirk Wilmuller and Robert Wayne, and is currently doing a postdoc at UCLA as well. That's right, Chris, right?
Yeah.
Cool. Awesome.
Still here in LA. They can't get rid of me just yet. Yeah, so I guess my general research focus is thinking about deleterious mutations, both in conservation applications, but more broadly in population genetics. To date, mostly my work on genetic rescue has happened just in this paper that you all read for this week, which yeah, I think was kind of motivated by thinking about how can we maybe better design genetic rescues. I think it seems to me that the paper's maybe been misinterpreted, at least relative to our intention, by a lot of people as saying genetic rescue is like bad. That wasn't, I think, what I had in mind when writing this paper, and I haven't read it in a while, but I don't think it really says that. I think it's more, again, like how can we, I think what the simulation showed in that paper is that genetic rescue generally has beneficial effects, but how could we maybe think about deleterious mutations and how they relate to demography, and how all of these things might help us design better genetic rescues. For me, I think a big open question in the field is how to go from a simulation where you can, have dominance and selection coefficients and fitness for all of your simulated individuals, and actually apply things like this in an actual population. One project I'm working on right now is thinking about this isolated caracal population, which is this wild cat species in the Cape Peninsula of Cape Town that's become isolated by the city, where they might, maybe there's still some ongoing migration, maybe not. They might perform a genetic rescue. If we want to then use genomic data to inform that, I think there's a lot of challenges that need to be sorted out. Yeah, and I think there's tools out there to try to tell us what, how much deleterious variation an individual has, but I think they're not that great, honestly, and I think there's a lot of room to improve it. So yeah.
Okay, great. Cool. Thanks, Chris. Okay, and then Sam Hauser, our last panelist, but not least. So Sam actually did her bachelor's at Rutgers, where I am right now. So not in this room, but just been in this building for sure. So went from Rutgers to the University of Louisiana, Lafayette, where she did her PhD. Did a postdoc with Emily Latch at UW Milwaukee, and I think has done a super interesting career move where she's now at Embark Veterinary. So they do a lot of the like canine genomics work. So yes, trying to kind of take her background in conservation genetics and genomics and apply it to our best friends, basically. So yeah, I'm super interested in hearing about that, Sam, if you want to give a little bit more info on that.
Yeah. So my background, like Brendan alluded to, was conservation genetics of in situ and ex situ populations. I worked with endangered species, just understanding population structure and connectivity. Of note, I worked with the Hawaiian monk seal, which has some of the lowest genetic variation. And then my other work prior to coming to Embark, which makes more sense of how I made the switch, was I worked with zoos and aquaria to incorporate genetic or genomic information into their breeding programs. And you can kind of see the natural link to genetic rescue there. Now I'm at Embark. I'm a research scientist here working on breed ancestry. But I also have a really cool component of my work where I get to consult with breed clubs to work on outcrossing programs to increase genetic diversity and inbreeding depression that they're seeing in their breeds. In terms of how I see genetic rescue, broadly, I agree a lot with what's already been said, so I don't want to be an echo chamber. But I do agree that it is one of many tools. Conservation is a complex and uphill battle, and we have to apply as many tools as possible. And I also think that one of the main issues we run into is data deficiency. We don't know for many species, their population structure. We don't even know their level of genetic variation, inbreeding load, or anything like that. And I think that is a major barrier to being able to do anything with genetic rescue. Did that cover all the questions?
Yeah. No, that was great. Cool, awesome. Well, thanks everybody. And I think yeah, now we can just I was just gonna put a couple of questions to you guys, and any of the other reading group participants have questions, feel free to ask them, either raise your hand or just unmute yourself and ask. That would be fine, like anytime. But yeah, I guess I'll ask just kind of just to start things off. So I think reading Chris's paper, there's definitely kind of an emphasis on the size of those source populations. So effective population size is definitely going to be an important determinant of load in a lot of cases. What else would it be helpful to know about either the source populations or the recipient populations in terms of their demographic history? Beyond just the current effective population, for sure, that would help us design effective genetic rescue programs.
Yeah, thanks for that question. Right, so yeah, in our paper we show that like source population size, that maybe there's like a sort of sweet spot of having like a moderate sized population that is to some extent purged of its inbreeding load for more thinking about like increasing the long-term efficacy of genetic rescue. You know, the first thing I would say is like the extent to which those populations actually exist in nature is unclear to me. Other things to consider, I don't know, I feel that there's probably a lot of practical considerations in terms of just what's there that, you know, that's not something we're thinking about in our very like academic simulation type of setting. Other factors are maybe, you know, just how divergent are these populations? Like to what extent are they adapted to differing conditions? I, at least at like an interspecific scale, and maybe less concerned about things like outbreeding depression. I know that that's like a big legacy in the field of like concerns about mixing populations that are too divergent. That's prevented much genetic rescue from happening, maybe. Doesn't really seem like that within a species at least is like maybe that huge of a concern, but also maybe that's a topic that deserves more research. And I mean, could be modeled, or, you know, experiments could, I mean, there's lots of experimental studies in that, but yeah, I mean, I guess those are the things I would think about. I'd be curious what others might add.
Is this specifically about recipient populations or source populations? Which one were you all wondering about?
Either, I think. So yeah, what would we want to know about? I just, you know, thinking in general about, you know, any kind of characteristics of the source or the recipient or the recipient populations that would help us to kind of better target these rescue efforts.
Yeah, one thing I've thought about a lot recently is, so just if you hold genetic load constant across a bunch of potential recipient populations, there can be vastly different population dynamics depending on which vital rates are affected by, well, depending on life history first of the species, and also depending on which particular vital rates are affected by inbreeding. You can have really strong inbreeding depression on one vital rate and no effect on population dynamics. And the same effect, or the same inbreeding load affecting a different vital rate and have a really big effect on population dynamics. So basically due to variation in sensitivity across different vital rates, or relative effect on mean absolute fitness. And, you know, I don't think we have a whole lot of information on, you know, how often, how common inbreeding depression is that has no effect on population growth. So like a lot of these populations that have been around for a really long time at super small effective population sizes have to have some level of inbreeding depression, because mutation is a constant in all species. And so in those cases, there must be inbreeding depression, but there's obviously not at least a constant effect on, or not a you know, an existential threat to those particular populations. And sort of understanding, you know, how common that is, and how common is this sort of soft selection versus hard selection, which affects population growth. Sort of understanding the relative frequency of those in the wild, and how often it changes through time and across space, through time within populations and among, or across space, sort of among populations. That's something I've been thinking about a lot lately.
Marty, could you give a biological example of what you mean by inbreeding depression affecting like some vital rates but not others?
Yeah, like strong inbreeding depression on juvenile survival, for example, can have very little effect on population growth in some species. But if you extend that effect into older age classes, it can have a much bigger effect on population growth. And another example, or another related issue related to life history, some species that have really high fecundity can sort of absorb a higher genetic load because they have sort of more surplus offspring, that, you know, the population size can be maintained at a large size, even if they lose a very large fraction of their offspring because of inbreeding depression. Does that make sense?
Yeah, absolutely. I mean, that's kind of what we see in oysters because they have really large effective population sizes, but it's also been estimated they have a big breeding load. So trying to get your head around that.
Any thoughts, Sam, on this? Or
A bit of the same. I'm often thinking about whether divergence of population really matters. I mean, a lot of genetic rescue is based off of trade-off of risks because we don't know a lot about their genome. And it's always a little bit of a gamble on whether that population divergence is going to cause outbreeding depression. I think the risk is low, but it's also an area we really haven't looked into enough either. I think about this a lot with now that I'm working with dog breeds, because breed clubs want to keep like the intrinsic or innate behaviors that they've bred into the breed. And if they outcross, is that going to maybe alleviate inbreeding depression, but ruin something really important about that breed? So we have to kind of balance that kind of outbreeding depression in terms of what the needs are with the humans. And yeah, I think it's important to also, in wildlife, we also have to remember there's logistic factors to all of this genetic rescue as well, right?
yeah, for sure. Yeah, it's let me maybe put another question to you, starting with you, Sam. Since you're working on kind of maybe not a model species, but a better study species than most of us. What can we, what are there things that we can do in these model species that we can't do in non-model species in terms of detecting deleterious variants and quantifying load? And, how would we develop better tools for doing that in non-model species?
That's a good question because we haven't really worked too much into it. This is kind of in its ideation, initiation phase of research. But one thing that we're talking about doing is looking at like runs of homozygosity between different breeds that we're thinking of crossbreeding to identify, and I guess this kind of gets at the functional genetic part, which I don't want to put too much emphasis on because there's not that much application outside of maybe model species. But we have been using runs of homozygosity and comparing that to other breeds and seeing if they have diversity in those areas, and we can break up inbred areas of the genome.
Cool. Any other thoughts on that? What kind of where our tools are lacking, or how much further this, or how much better we can predict the effects of or how much better we can or can't predict the effects of a given kind of management action if we have a reference genome, or if we have a good reference genome, or if we have like annotated variants or something like that.
Yeah, I guess I mentioned before, this is something I've been thinking about, that to some extent, I feel recently the field has been putting the cart in front of the horse a bit with like using a lot of these tools like GURP scores or CAD scores or whatever to like say that like we're using them to estimate genetic load. but like, again, they're not giving you a selection coefficient. And I mean, I think there's some studies out there that show that like a GURP score, which for those who aren't familiar with one, is like the score that's kind of based on evolutionary conservation. So you align a bunch of genomes and look for sites that are really conserved, and like assume that if you have a mutation at a super conserved site, that it's like highly deleterious. But I think there's some evidence to say that like a method like that can kind of tell you what's probably deleterious from like not deleterious at all, but it can't really like tell you what the really strongly deleterious stuff is. And nor I think can any other methods out there right now do that very well. And the other big lacking component is dominance. Like if we're thinking about anything having to do with inbreeding and inbreeding depression, like we need to know what the dominance coefficients of these mutations are. And I think that's just like, in general, in evolutionary genetics, like a topic that we have woefully little information on, because it's just a really hard thing to do, to like estimate dominance, like genome wide, let alone for like a single mutation in your VCF. So yeah, in that sense, I mean, I like what Sam mentioned, like focusing on things like runs of homozygosity, I think are a bit more like unambiguous to me. Like if you see a big chunk of the genome that's homozygous, like there's a pretty good chance that there's like recessive deleterious mutations that are in there that are exposed. Which ones they are, or like where they are, like who's to say? But yeah, I think there needs to be a bit more skepticism about some of these tools that are out there for assaying deleterious mutations. Yeah, Katie, you have a hand up?
Yeah, I was wondering if you guys had any experience with these programs I've heard about, but I haven't used, where they're like trying to estimate the like how deleterious a mutation is based on its change in the amino acid sequence. Like if it changes from a hydrophilic to hydrophobic or something at that amino acid. Are you familiar with those at all, and what's your opinion on them?
Yeah, I haven't worked with them at all, but I think that's maybe where things are heading, is like some of these, I think one of them is called like Eve, right? It was like this big Nature paper a couple of years ago that had this big machine learning model to like try to predict protein folding and get like better scores of mutation fitness effects. I think maybe that's where like this field should head, is like using programs like that and trying to apply them in conservation settings, because those seem to work pretty well, but I don't think they've been used much, if at all, yet.
I guess that, yeah, question. I guess they probably, yeah, I wonder, like they probably do a pretty good job of predicting changes in the function, but then going from there to like fitness is another like big question of how do we make that leap. Yeah. Any other thoughts?
I'll just say something. Sorry, Marty.
No, go ahead. Yeah, go ahead.
I just wanted to say, I have not personally done this, but I know smarter people at Embark have done a lot of this deleterious work, but only looking at kind of a gene of interest. And I've seen a lot of what Brendan has alluded to, of it's easy to identify them. It's really hard to then take that to the next level. And the rate of discovery that we have with the resources of a corporate corporation and a large science team is pretty slow. And so I wonder if that application is possible in conservation, where we don't have the same sort of resources or reference genomes.
Yeah, I was going to sort of reiterate that and maybe take it further. You know, what we're interested in evaluating population viability is essentially trying to estimate population growth rate. You know, is a population likely to grow in the future, or is it likely to decline in the future? So what we need to do is estimate the impacts of something of anything that we're interested in, any sort of threatening factor on vital rates, so age and sex specific survival and reproduction. And I am very skeptical that we're ever going to be able to try to identify deleterious variants in sequence data and then reliably convert those into vital rates. And then, to then build a population model where we can, based on that information, reliably predict population viability.
All right, this is awesome. This is a good conversation. I think maybe since we're a small group, I think maybe now would be a good time if we want to go around and have some of the folks in the audience introduce themselves. And if you have a question, feel free to ask it. That would be great. But yeah, we can just kind of go around on my screen. Nicole, if you want to introduce yourself.
Hi, everyone. My name is Nicole Collars, and I am a postdoc at Northeastern University with Randall Hughes. And I'm really interested in questions of interspecific diversity, the causes and consequences of interspecific diversity, and how we can apply knowledge of that to restoration efforts. So genetic rescue is, of course, a really hot topic in plant restorations right now. I'm working with a project that's considering bringing genotypes from southern United States up here to New England to help improve salt marsh restoration. So I'm still new to the field, but I've really enjoyed hearing your thoughts and reading your papers, and thank you for being here.
Sounds like cool work.
Cool. Sarit, do you want to go next?
Hi, everyone. I'm Sarit. I'm a PhD student at Northeastern, also in the Hughes Lab. And I've been working with Katie a bit this semester too. I'm broadly interested in the integration of genetic and genomic data and tools into conservation and restoration. And for my PhD, I'm thinking about that through the lens of oyster restoration up in the Northeast, where oysters in the wild are still both super low density, and the thinking is genetically depauperate. And so I'm just kind of doing some work with wild populations and a bunch of restorations. And yeah, excited to be here. Thanks.
All right, Nicholas.
Hi, I'm Nicholas. I'm currently a postdoc at UC Berkeley. So for my PhD, I was working on the population genetics of Atlantic cod and some technical aspects of low coverage whole genome sequencing at Cornell. And right now I'm working on the sort of rapid diversification of rockfish and also structural variant discovery and analysis with long read sequencing data. I really enjoyed reading both of your papers, and thank you very much for joining the panel. So I guess I do have a question, which is given what Marty said about the importance of really tracking the demography and vital rates of the population in question, I'm wondering how important it is for us to have neutral genome-wide genetic diversity as a central metric to assess the health of a population, or as a metric to sort of for conservation effort, or to decide whether a population needs to be rescued or not. Or I guess on the other hand, whether it is worth it to devote some resources to look at the so-called adaptive genetic variation.
Is that to me?
To all the panelists, I guess.
Oh, okay. Well, the first thing I'd say is that I think looking at sort of genome-wide, neutral, putatively neutral genetic variation is informative. So if you look at a range of populations and one has lower heterozygosity than others, you can infer that that population has higher average inbreeding than other populations. And, it's useful to look at the general population trend. So is the population stable demographically? Is it declining? Is it failing to recover after protections have been put in place? I think those are two key bits of information. So levels of genetic variation, and then, is the population growing, declining, or remaining stable? Would you expect it to be growing given the present environment and protections put in place? Are the vital rates, if you have more detailed demographic information, that's quite useful too. Or, is juvenile survival much lower in that population compared to others? Those sort of can point one in the right direction, I think, in terms of whether genetic rescue is needed or not. And, I agree with Chris, sort of these genomic metrics of genetic load are interesting, but we really don't quite understand what they mean yet in terms of translating them into fitness. And I think those sorts of analyses are always interesting. And, from an evolutionary standpoint, we can study, those mutations have to be, these mutations that are conserved and mutations at sites that are conserved or at protein coding sites in the genome. We know that on average, those should be deleterious. So we can look at variation at those sites and learn something about how deleterious genetic variation has evolved, but translating that into demographic information is difficult. Please chime in, Chris and Sam.
Yeah, I think I'd largely agree with what Marty said. I'd maybe tend to have a slightly differing view on the relationship between genetic diversity and genetic load. I think, it sort of depends on how if you're going to be low diversity, it sort of depends on how you get there. If you get there gradually, like maybe you're going to be in much better shape than if you get there very suddenly and rapidly. And yeah, I think that's where, I don't know, it's challenging. I think in conservation genetics and conservation, maybe in general, we want kind of like simple universal rules for managers to understand and implement. And yeah, maybe in most cases, a simple relationship between diversity and something like fitness exists, but in a lot of cases it doesn't. And as an academic, those cases are interesting to me, but like, is that going to be too complicated for a conservation manager to understand? I'm not really certain, but yeah, I think it's just the nature of what we do, like in any field in biology, but especially ones that are integrating like very different complex fields, ecology, genetics, evolution, like things are complex, and trying to find like the right balance between like complexity and like simple general rules, I think is always challenging.
I agree with both Marty and Chris, but the only color I'll also add is I think if there was structural variation that was pertinent to what's going on with the species, and that's going to be on a case-by-case basis, like most conservation is done, then potentially we could use it. But logistically, most species of conservation concern don't even have a reference genome. And so we won't even really have any foundation to look at structural variation, unfortunately.
Great. Yeah. Cool. Thanks, everybody. Let's go around quickly, let's try to get everyone get introductions from everyone. I don't know, some people have to leave at the top of the hour, but let's just see. Joanna, do you want to introduce yourself briefly?
Sure, yeah. I am Joanna. I think I'm frozen. Hopefully people can still hear me. Yeah, okay. I'm a postdoc at University of California, Davis, working with Andrew Whitehead and Nan Fungi. And I really enjoyed the conversation today, especially because UC Davis just had our meeting group today to discuss some of the papers, and we were talking about ways to look at genetic load and how it seems like it's, I don't want to say like outdated practice, but it's something that was sort of came about before sequencing of and yeah, just really interesting to think about the ways we measured that in the past and if there's any way to do it with any type of whole genome sequencing data now. And so I thought this was great because you guys literally answered some of our questions we talked about today. So I was also sort of wondering if it's possible to do that with like a GWAS type study, if you could do survival and then think about it that way. I'll sort of pose as a question, but then I also wanted to just quickly jump in about some of the research that I'm currently doing, which is with delta smelt fish, which I think is going to be a really interesting example here in the future because they are now extinct in the wild and they only exist in a hatchery. And they've been doing a breeding program for about 10 years and they've genetically managed them. And so there's a pay, and so they only cross fish with very low relatedness, which I think has been really great at like conserving, thinking about whole genome genetic diversity, but I don't think anyone has looked at, like they've only ever used like a 75 SNP panel. So no one's looked at whole genome diversity or like genetic load. And I'm so curious to what's going to happen in the future as we now only have what's left in the hatchery. Yeah, so this all been super relevant.
Cool. Yeah,
I mean, I'll chime in quickly. I don't know GWAS necessarily how that fits in, but maybe related. One thing I've been thinking about for a number of years now that it's a general type of study that I think would be challenging to pull off, but would be cool on this topic is just being able to track individuals at varying life stages in a population and have genomic sequencing data and kind of see how allele frequencies change with time and use that as a way to look for highly deleterious alleles under selection that might be enriched in, say, like juveniles, but not present in adults. There's one paper I can think of that from a few years ago that tried to do that in humans, but largely had samples from like older humans. So maybe is missing out on all the interesting stuff that could be happening early on. I don't know how much like conservation relevance a study like that would have, but I think as our ability to generate large genomic data sets continues to grow, like having really fine scale, large sample size studies of selection dynamics in natural populations will be more and more feasible.
No, I just add a little to that. There are methods for using data on runs of homozygosity to map loci contributing to inbreeding depression. There's a nice example in soy sheep. Martin Stoffel's paper in soy sheep, they map several loci that look like they contribute to inbreeding effects. There are also a few examples where some really large effect loci have been identified in basically where lethal mutations have been detected in natural populations. There's a couple in birds. There are mapping approaches that can be used, and in addition to the approaches Chris was mentioning.
Okay, thanks guys. Yeah, Nicole I think has to leave now, but she's in the Whitehead lab too. I'm just gonna read, she wrote a little bit unless you've got time to just say a few words now. But
I can stay on for one more minute. Yeah, I just wanted to give my quick intro. Also in the Whitehead lab with Joanna. So I'm in a pretty different field from this, environmental toxicology. I study multi-generational effects in Atlantic killifish from exposure to flame retardants, primarily focusing on fish behavior and brain gene expression. And I mostly joined the reading group so that I can kind of broaden my perspectives and learn more about evolution and conservation genetics and the different tools to use. Yeah, I also want to say thank you for the panelists. This was really interesting to listen into. So thank you.
great, thank you, Nicole. And Danny, I think you're our last. We haven't introduced you yet, but I'd like to hear from you.
Yeah, hey, thanks for the panel. It's really good. Yeah, I'm Danny. I'm actually in Halifax right now, and I'm in Ian Bradbury's lab. I'm a postdoc, and I joined the RCN because I joined the reading group originally, and then I started working on the Marine Omics website with Catherine and Jason and Katie and all them. So that's how I got involved in this. And yeah, this is really interesting to me. I actually work on copepods, so they're not going to be in need of genetic rescue anytime soon. So I'm working on using like genetics and species distribution modeling and stuff to kind of understand where whales are going to end up. But I have read a lot of your papers, and I've been really enjoying these panels and going through the reading group because I'm also part of a project called STAR, which is a project where we're reintroducing extirpated sharks into Raja Ampat, which is a shark sanctuary in West Papua. And so, yeah, we did a bit of genetics, but yeah, we're just sort of keeping an eye on it because essentially there isn't any sharks there. So we're just kind of, I am interested in this because these populations of sharks are pretty small to non-existent, and we're just basically moving sharks from other areas into there. So it's already happening. So the train cannot be stopped, but the geneticists are sort of like keeping an eye on it, and I'm one of them. So yeah, really interesting.
Yeah, that's something that I was thinking about, and if people have to leave, feel free, but just kind of thinking about thinking about how you would, or just what relevance genetic rescue has in marine species, which a lot of the folks in the RCN work on. I feel like a lot of the examples are in terrestrial species or freshwater species. And we kind of often think about marine species as, being, like copepods or having huge populations, and they probably don't need it. But then things like, Marty's working on orcas, and Sam was working on the monk seals at some point. So yeah, what do you guys think? What are the lessons for kind of marine species conservation in this?
Brendan, can I tack on to that as well? So just to tack on to that question, we're talking like whales and seals and some sharks. Those are very different than plant and oyster populations, for example, that a lot of the people in our group study. But genetic rescue is becoming increasingly acknowledged as a possible way to help restore ecosystem function in oyster and plant populations. So we're not going to just bring one oyster or one plant into a population. It's going to be like hundreds of plants or hundreds of oysters that we might bring into a population in a rescue effort. So I'd love to hear your thoughts, especially Chris, how those models might change from your paper when we think about bringing lots of different individuals into a population as a genetic rescue effort.
Yeah, that's a nice question.
And sorry if I sidetracked us, Brendan.
Go ahead.
Oh, I was just gonna say, sorry if I sidetracked us, Brendan. I didn't mean to take away the glory of his question, but another layer to add.
Yeah, I guess I haven't spent a ton of time thinking about organisms like oysters and how dynamics might differ. I think in general, if you're bringing in more individuals and, you know, the recipient population is in need of that, that's probably going to help even more, even just on a demographic side. The broader question of how these factors apply in marine systems, I also generally tend to see the world through the terrestrial systems that I largely study. And I don't know, I'd be curious to hear others weigh in on, like, I mean, what would it even look like to capture an orca or a shark or whatever and move it around? Like, is that something people have done and feel like they could do? I don't know.
I can see, like, with this STAR project that I'm working on, where the species is Stegostoma tigrinium, which we call the zebra shark, and I think you guys got the leopard shark. But either way, they can be raised in hatcheries, and so we just got a bunch of them from places that were kind of close. That was kind of the approach we ended up taking. There's not many of them, but the ones that we did have, we tried to get populations that were as close to the location as possible. And yeah, they're raised in hatcheries, and then they fly them out and release them into the wild.
Cool. Yeah, very nice.
With the monk seals, there's quite a bit of translocations across the islands. But I guess the monk seals are also not a great system to apply to these other marine systems because they're quite unique in that they have extremely low genetic diversity. I think it's like 3% heterozygosity, something obnoxiously low. But their populations are increasing. But a lot of the reasons for the translocations are also for social reasons in terms of hierarchy and aggressive males killing babies. And so like there needs to be translocations rather than for a genetic rescue reason. Yeah, sorry, I don't know if that's super applicable, but there is work being done in terms of translocations, and they're not necessarily done for the reason of genetic rescue, but genetics are considered because we don't want a risk of outbreeding depression or anything like that.
Yeah, with killer whales, people have captured many, many killer whales. I mean, all of the killer whales that are in SeaWorld are there because they were captured from the wild. In terms of moving them around and translocating them, we've thought about this a lot, and we don't really think it's possible. You know, if you captured a killer whale in Alaska, for example, in a bigger, more genetically diverse population and translocated it to the Puget Sound, where there's a much smaller population with low genetic diversity, that whale would probably just turn around and go home. You know, the whales we work with have massive home ranges, and they're isolated by social structure more than by barriers to gene flow. You know, they have these really close-knit matriarchal lines, and they speak different languages, different populations do, even within ecotypes. So they're, you know, there must have been some genetic connectivity among these populations historically, because they still have pretty high genetic variation. Well, in a relative sense, they do not have extremely high inbreeding. What was I getting at? So the point is, I don't think genetic rescue is really a, you know, viable management option for something like killer whales that just have these massive home ranges, and they interact with each other, individuals in different populations all the time. They just don't breed with each other. Very often.
Can I pose a hypothetical question? So let's say you were in charge of or advising a restoration effort that is starting from a blank slate, and you get to choose the genetics. You get to choose where you source the population, source all the individuals from. You get to make choices about diversity. You get to make any choices you want. You're designing the genetic makeup of that population. What would be your top consideration to prioritize, especially, let's say, the oceans are getting warmer, or sea levels are rising, and the salt marsh plant is going to drown if in the next 100 years or so? What type of factors would you prioritize in making those decisions?
I can speak to this because the monks, it's kind of in the reverse direction though. We drafted an emergency breeding program that would go into zoos in case something horrific happens. And essentially what we did was we were trying to minimize risk for the future, which means maximizing genetic diversity and variation. So there's a few rules with that, with ex situ breeding programs, which Joanna probably already knows a few of those things, right? You want to get, you want to be representative of all the different population structures or different populations you might have. You want to maximize breeding potential. So even males to females, if that's the breeding, right? That's going to change based off of the species a little bit. And you're going to want unrelated individuals. So there's a few kind of rules of thumb that you can employ when doing this. But if this were like if this was pulling that into a new area that was extirpated, I would follow a few of those rules, but maybe potentially you would want to consider or favor populations that were close by so that they may be locally, there may be that local adaptation that you don't know about and can't account for. And they might have the easiest ability to kind of work into that system.
Yeah, I think that's a really nice answer. I don't know that I have a ton to add. To me, I think the one important component that I also think is worth emphasizing when thinking about like what kind of motivated our paper or simulation paper is a big thing I think is like if the population you're thinking about has much potential to grow. Like some populations I think are just destined to be small by the nature of being a island wolf population, or our mountain lions here in Santa Monica, the Santa Monica Mountains, like they just can't support more than 20, or maybe for the Isle Royal wolves, 30 or 40 individuals. And to me, that's where like thinking about more carefully about these kind of deleterious variation things and inbreeding load things becomes more important, because in a case like that, inbreeding is pretty inevitable. And I'm also not certain that like maximizing adaptive potential. I have a hard time imagining a ton of adaptive evolution happening in a population where you have an NE of like 5. And I don't know, maybe that means a population like that is sort of doomed from a adapting to climate change perspective. But I tend to think of, these severe effects from inbreeding depression being the more like imminent threat. So yeah, but all of that kind of goes out the window if you're thinking about populations that have lots of room to grow and aren't inevitably inbreeding and can sustain higher levels of diversity and potentially adapt to things.
Yeah,
Nicole, you're talking about a scenario where you have basically an empty environment and you're reintroducing a species to an environment. I just wanted to make sure that's the case.
Yeah, for example, let's say there was some development, coastal development that destroyed some salt marsh, and one of the conditions for that development was that development group got in touch with a environmental consultant group to build a marsh somewhere else. And so you're starting from scratch. You just identify a piece of shoreline and you're going to build a marsh there with the hope that it's going to stay and it's going to persist. And so I think a lot of decisions that are made is just get plants in the ground and prioritize biomass. But if you have an opportunity to design the genetics of that population, what would you consider?
Okay. Yeah, I think I agree with most of, well, basically everything Sam and Chris have said. And I would maybe add that, thinking about whether, well, I think Sam actually mentioned trying to get individuals from populations that are close by, hoping that they would be locally adapted, and making sure that there is genetic variation, a substantial genetic variation among individuals. Like, you wouldn't want to take all the individuals from one SIB ship and put thousands of them in the ground.
Cool. Does anyone else have any other questions for the panel?
I don't have a question, but I just want to say this has been a really, really good discussion. I learned a lot, and there's another project going on in my lab where I think a lot of the things that you guys have talked about is going to inform what we do. So I'm really glad that you all came today.
Thanks for having us.
Yeah, thank you. Thanks for organizing this, Brendan.
Yeah, this is great. Thanks.
No problem. Yeah, thank you, everybody. Yeah, and hopefully we'll have another one of these happy hours at some point for one of the next sets of readings. I think we're going to be contacting some other authors of some of the other papers. So yeah, it's in the works. We'll let you know when it happens. Cool. Thank you, everybody, for coming, especially the panelists, and yeah, hope to see
Good seeing you all.
Yeah.
Bye.